Okay, we'll go back over. Here we are. Okay, so we are at this polypeptide. There we go. Polypeptide product. And the polypeptide is, as you guys are hopefully aware, that is the primary and secondary arrangement of a protein. This guy can be further modified through something called post-translational modifications to form the protein or enzyme final product, right? Future, whatever that is. And this is called post-translational modifications. This means, if you recall from our chapter 7, when you have those internal like disulfide bonds or phosphate bonds or hydrogen bonds that are formed within that polypeptide string to form that tertiary structure, and then when those subunits get together to form that quaternary structure of that protein, that's what we're talking about, modifications. Okay, this translation process here where mRNA is translated, and this concept translation, just think like when we use this term translation, you're translating like English to Spanish or something, or vice versa, right? You're translating something that has the same meaning from one form of language to another. And that's what this translation does here too. It takes the information in ribosomal RNA form and just changes the form of that information and still keeps the information the same, but it's in a different form. In this case, it's amino acids, right? So polypeptides are amino acids, if you guys don't remember. This is that sequence of amino acids generated from information that's in that mRNA transcript. And the machinery involved in translation are all those that you see in this picture here. So we have our ribosome or ribosomal complex, which has both the ribosomal RNA this is the small subunit. Of this complex here. And then we have the large protein unit, right? This is the large subunit of this complex. And then we have these really key intermediates here called transfer RNA, tRNA, or transfer RNA, transfer RNA molecules. And their job is to bring in amino acids, match them perfectly with mRNA sequences, and form that polypeptide sequence there. So the ribosome, the transfer RNA molecules, and of course the key ones are amino acids. So this is the process we are walking through right now, is protein synthesis from a gene transcribed into mRNA and translated into a polypeptide product. Note that transcription occurs not just for that gene region, but also all of these RNA units, all of them come from DNA also from different regions. So that ribosomal subunit and that transfer RNA, those transfer RNAs all are from different regions of the DNA, and transcription produces all of them. Also, just as a side note, so you're aware, this ribosomal RNA, the gene segment that encodes for that ribosomal RNA, is actually how we get something called, what we do, 16S bacterial identification. So this is how we get identification of bacterial species, is by actually looking at the sequence of the 16S unit of the ribosomal RNA, which is that small subunit that makes up that ribosomal complex. And this allows us, just by doing this genetic analysis of these regions called the variable regions within this ribosomal RNA DNA unit, is enough to differentiate one organism from another in the entire bacterial kingdom. Kind of cool, just so you're aware. Okay, so let's look at this process of transcription and then translation. And also, just so you know, there are other types of RNAs that can be generated from transcription. These are all involved in regulation of either metabolic processes or even gene expression. We're not going to talk about any of those here. We're really just focusing on transcription of mRNA, but be aware all of this stuff is occurring at all times. Okay, so transcription here, again, we're going to kind of keep it simple. We're now working with RNA instead of just DNA. RNA is a single-stranded linear molecule. It is similar to DNA in its core structure, so it still has that sugar phosphate backbone, but instead of deoxyribose sugar, this has a ribose sugar. The big difference here is that 2' carbon. It's hydroxylated in ribose, and there's no hydroxy group here in deoxyribose for DNA. There is a nitrogenous base still connected to that 1' carbon, just as we saw before. And these are the 4 base combination nucleotides that can be at any of these sites for the nitrogenous base. There, of course, is adenine, guanine you're familiar with, cytosine, and now instead of thymine, we see uracil. So this is the key, one of the key differences between DNA and RNA is the replacement of thymine with uracil. Just so you're aware, the rule of base pairing is actually quite similar, but we'll see for transcription, what we're actually seeing paired is from a DNA template to an RNA transcript. Sorry, RNA transcript. And so for DNA, when you have a cytosine, this will base pair with a guanine still for RNA. When you have a guanine for DNA, when you have a guanine, this still will base pair with a cytosine. When there is a thymine in DNA, this will base pair with an adenine in RNA. But now is the big difference when in DNA there is an adenine, this will base pair with a uracil. So this would be the rule of base pairing when we are talking about transcription. Okay. Again, the key differences here is that we have a ribose sugar instead of a deoxyribose sugar for RNA versus DNA. Uracils replace thymines for that nitrogenous base. And then RNAs are single-stranded and linear versus DNA is double-stranded and circular. Those are 3 key differences between DNA and RNA. That might be on the exam. Okay, let's look now at this process of, well, this is one more picture to show you the differences. There's a single-stranded RNA, double-stranded DNA, and just so you're aware, these nitrogenous bases are highly reactive, just like we talked about for DNA replication. So these guys don't actually stay in this open, exposed form. They always fold back in on themselves to form these really unique structures or have chaperone proteins that bind to help them stay in this linear form. So they don't just kind of hang out like this in the cell. And here are cytosine, guanine, adenine, and then here's uracil in the place of thymine. And just take a look, these molecules are actually quite similar in their structure. There's a methyl group here on thymine that's not here on the uracil, but otherwise the binding is very, very similar. So this allows the properties between these 2 molecules to be very similar also. Okay, here is the process of transcription. So as mentioned, and I know I mentioned this in videos, so forgive me if we haven't actually face-to-face talked about this, but let's just remind ourselves. For prokaryotic cells, actually for all prokaryotic and eukaryotic cells, here's our chromosome, double-stranded DNA molecule. As mentioned, we have these gene segments here. For prokaryotic cells, usually these are 3 or 4 different genes that are collectively regulated as operons, right? So I'll just say operon. Now for all of these gene segments, and this is true for eukaryotes and prokaryotes, the cell needs to know when to begin reading this and turning it into a protein. So for every gene segment or operon for prokaryotic cells, there is a region that's at the start of the gene that indicates the start of transcription. And this is called the promoter, or P. This is the start of transcription. Because this is the docking station for RNA or RNA polymerase, okay? So RNA pol binds here or docks at the promoter region. So you'll find this at the start of every single gene or gene segment on both prokaryotic cells and eukaryotic cells. And this is not actually it's not a gene, it's just a sequence. Oh my gosh, sorry guys. A sequence of nucleic acids that is consistent and is essentially a binding site for one of the regions on that promoter molecule. Now, in addition, just because we'll talk about this next week, and because we talked about it in the lab portion. There's also a region here called the operator. Sorry, I think I was writing it in blue before. Let's just keep that consistent. The operator is also located right at the start of these gene segments, not on every single one, but on most of them. And the operator is important for regulation. So the operator is involved in regulation. This is like the on-off switch, essentially, and it's the docking station for a repressor molecule. So we'll talk about that more in our next lecture discussion when we're talking about DNA regulation, and also we'll talk about it more in the lecture, in the lab portion. But this is usually sandwiched between that promoter region and the actual structural genes here in this region of the operon. Okay, now we can go back up here and look at this simple process. Number one, an RNA polymerase recognizes and binds to the promoter. So it sits and docks right there at the start of the gene. Once it does, it elongates, so it creates that mRNA transcript. And note that the direction is exactly the same as we talked about for the DNA polymerase from before, right? Transcription always generates that new strand from the 5 to 3' orientation. Okay, so the new strand is always made from 5 to 3 because of the same rule that we talked about before, that 3' carbon must be available for the addition of nucleotides during this elongation process. This process will occur all the way until a terminator is reached at the end. You can add that to your little chart over here, that at the very end of the gene or operon, you'll have this sequence of nucleotides that is your terminator, E, right? So this indicates the end of a gene segment, and it initiates the termination, so everything essentially falls off of the process of transcription at that point. Now let's take just a second here and remember about strand orientation. Because we know that the new strand is made from 5 to 3' orientation, that means the template must be complementary to that, or 3 to 5, right? If you were to look at this double-stranded DNA molecule that we have here, the red and the blue, which of these strands will be the template for this RNA transcript? Is it going to be the red one as the template or the blue one as the template? Because only one of them will be used. What do you think? Okay, stop. So again, let me just ask you again. So the elongation or the new strand is made from 5 to 3. The new strand's orientation is 5 to 3. So considering this, that the new strand is 5 to 3, what must be the template, the thing that starts off this process or is read by the RNA polymerase? It must be the complement to this. So which of these two strands, the red or the blue, is complementary to a 5 to 3' orientation? There we go. Yes. Nope. They're the blue, right? So we're looking for the template to be 3 to 5. And if you look at these two strands, the red is 5 to 3 and the blue is 3 to 5. So in this example here, the blue strand is our template, right? The blue strand is our template. There's some terminology you can use. I'm not going to test you on this, but if you notice here, the template is called the minus strand of DNA. The other strand is called the plus strand. It's also called the coding strand. So if you see that terminology, and it's really because if you were to look at the sequence of the non-template strand, the sequence is exactly the same as the mRNA transcript, with the exception that the thymines and the uracils are changed, right? It's not opposite, but complementary, Jeff, yeah. So the template strand is always complementary to the mRNA transcript. So here we have the transcript in 5 to 3' orientation, so we know that the template is in 3 to 5' orientation, always complementary, okay? So here we can see during this just shorthand understanding, if you had this double-stranded DNA molecule, identify the template, and then for transcription, we know that there is that rule of complementary binding that we just mentioned before. Thymines are complementary, or adenines are complementary to thymines. For adenine here, the complement in RNA would be uracil. For the cytosine, the complement is guanine. For this guanine, the complement is cytosine, right? And you would continue reading essentially all the way down, reading this template and then transcribing it into this mRNA transcript. Now for conceptual understanding, let's look at the next slide. Let's look at the next one because this one's better. So for here, you see that RNA polymerase actually works a little bit, similarly to DNA polymerase. It's a big fat enzyme and it sits on a wide region of that DNA. But unlike where we talked about DNA replication, where that double strand opens up wide and both of those act as templates and they'll stay bound to their new strand, here we have a very small region of that DNA that opens up only where that RNA polymerase is sitting on that double-stranded DNA molecule. And as it moves along, essentially it opens up that double-stranded DNA molecule only so that the template could be read and an mRNA transcript formed, but it continues to read along. And as soon as it passes by a small section of DNA, the DNA will just re-anneal, close right back up again, because DNA has a stronger preference or affinity to itself than it does to mRNA. So that mRNA transcript is just kicked off. It just stays kind of tagged along that RNA polymerase complex as the transcription is occurring until that polymerase hits that terminator, which is indicated by that red coloration here. That terminator is just a signal sequence that terminates this process. The RNA polymerase will disassociate. It will also kind of kick off that mRNA transcript, and there's something called a hairpin loop that's formed here that terminates that whole process. So this is just, it's important to note that that double-stranded DNA molecule, nothing changes. It just opens up temporarily to be read and it closes right back up again. So because of this, that same gene segment can be read over and over and over and over again without ever being modified during this process. In the end, you'll have that mRNA transcript, but remember that there are other types of RNA, including transfer RNA and ribosomal RNA that can be generated from a transcriptional event, and all of those ingredients will come together for the process of translation. So before we get to the next section, I'm gonna have you guys work on this for a second. This says transcribe the following double-stranded DNA molecule. And so the first thing you have to do is determine which of these guys is gonna be the template. And then from there, use that template to write out that mRNA transcript. And go ahead and write this down on your own, on a computer word discussion thing, or on a sheet of paper. I'll give you just a minute to work through this one, and then I'll ask you for your input. Be prepared, because I'm going to call on someone to unmute yourself and write and tell me what this is going to be. All right, hopefully you've gotten through enough. Let's see who's actually on here. Beverly, do you think you can help answer this one?
Yeah.
Okay, cool. Let me get a pen first, and then I'm going to have you read it out loud.
It didn't matter which one was the template strand, right? We could choose either or.
It actually does matter. So that's one of your jobs, is to choose which of these things will be the template strand. Which one did you choose as your template?
So if it replicates always 5 to 3, then I guess I would choose the bottom one as my template.
Yes, good. Well, shoot, sorry. Yes, this bottom one is going to be the template, because exactly right, because of the orientation. So for your mRNA transcript, what would you indicate as the start? Give me the orientation first.
5 prime.
And then what would it be?
C A U G C A G U A C U G G U, and then 3 A's.
Perfect. And then how would you end it?
And 3 prime.
Perfect. Thank you.
Mhm.
Okay, guys, keep this jotted down. We're going to use this exact same double-stranded DNA molecule in our example for translation too. So if you didn't get this, make sure you're kind of going over that again. Any questions on this one? We're okay? Okay, let's keep moving forward then. So translation is the process by which that mRNA transcript that we just saw generated is essentially converted into an amino acid sequence. And it's based on the sequences of these nucleotides in that mRNA transcript. So let's go over this concept of these codons real quick. And I didn't want to spend too much time on this, but oh, we have time. We're good. So first we have to understand this concept of the genetic code, and this is based on this idea of codons. So a codon is any sequence of 3 nucleotides in a row. Any 3 nucleotides would be considered a codon. If you were to do a statistical calculation of those 4 nitrogenous bases in groups of 3, you would find that there's 64 possible combinations. So there are 64 codons for both mRNA and for DNA. Of course, it's different. The T's and the U's are switched, but otherwise those groupings are the same. So here is a picture of sort of how this conceptually looks for the DNA. You can talk about codons in DNA. We usually discuss codons as a form of mRNA or anticodons as part of tRNA. But you can arguably say that DNA can be grouped in these sequences of 3 nucleotides in a row. So in this picture here, you'll see the first 3 nucleotides is considered codon number 1. After the process of transcription, the first 3 nucleotides in that mRNA transcript is codon also number 1. And then if you were to look this up on something called a genetic code or an mRNA codon chart, this particular codon is always associated with one specific type of amino acid. And you could do this all the way down the road for all 64 possible combinations of these groups of 3. All of them have a very specific amino acid associated with them. And this is called the genetic code, right? As mentioned, there's 64 possibilities, but there's only 20 amino acids that any organism has, with very little exception. So because of this, we call the genetic code redundant because there's so many possible combinations of those 3 nucleotides in a row. And let me show you a picture of what this genetic code looks like, or the chart. You can see lots of variations of this, but the actual encoding is exactly the same no matter which chart you look at. This is one from your textbook. Whoops. So just so you know how to read this, again, this is just looking at all of those 64 possible arrangements of nucleotides in a row. And this is mRNA, as you can see by the uracils. And to read this chart, the first base position is indicated on the left-hand panel. The second base position is on the top panel, and then the third base position within each of these different rows is indicated here. So for example, the start codon for every translational event is AUG. You can find that by thinking AUG, the first base position is an A, the second base position is a U, so we're looking right here in this box, and then the third base position is the G, so we're finding it right here, AUG. This codon always initiates a translational event. This is called a start codon, but you'll also find this kind of peppered throughout a translational product because methionines are just other amino acids. But importantly, you'll always look for AUG as the start of a translational product. Also, a couple of other things to note here. So note up here on the top of this codon chart, UAA and UAG and UGA. You don't have to memorize these. You'll always be given a codon chart to do these problems. But these 3 particular codons, or groups of 3, encode for something we call a stop. This is the termination of a translational product. So when that mRNA transcript is read all the way through, the very last thing that the ribosome machinery will sit on is one of these stop codons, and that terminates the whole process of translation. And as far as the redundancy, note that, for example, proline here, there are 4 different codons for series 3 nucleotides in a row. All 4 of these encode for a proline or are associated with a proline. So again, this is called redundant. Redundant means like it's repeated over and over, or the same thing is said twice. I was just being redundant. So here's a picture and evidence of that here. Serine, the same thing. Almost every single amino acid is encoded by multiple codons, right? With very little exception. Tryptophan would be the only one, and methionine. I'm sorry, those are the 2 exceptions to that rule. Otherwise, everything else has multiple codons that encode for the same thing. Keep that in mind, because when we talk about mutations in just a little bit, you'll see that because of the redundancy of these amino acids and the codons, there are some mutations that actually have no effect on that protein product, right? So we'll keep going. The last thing to mention here before we actually look at the process of translation is this concept of reading frames. Now, if you've already watched the video, you'll see me, I have already done this before, but I'll do it for the, for those of you who haven't seen my video yet. A reading frame you should be familiar with, just because we know how to read the English language. But let's say I had something like if this was like a sequence of nucleotides, right? And I said that we read, at least in our sentences, we read in groups of 3. And I said, group these in groups of 3 to decipher the sentence. Well, if you start at the beginning and you tried to group this in groups of 3, this would make no sense at all, right? The reading frame gives you a nonsensical statement. So the same thing is true with mRNA translation, that you need to figure out where to begin reading these groups of 3, or where to begin grouping the nucleotides in groups of 3 before you can actually decipher this into an understandable product or into a protein product. So for you guys, you know that in the English language, we begin reading with capital letters and we end a sentence with a period. And so in that case, you could say, well, I'm going to essentially just discount everything that comes before the first capital letter. So all of these things will be disregarded. And I will begin reading, my reading frame will begin there, and I'm going to group my letters in groups of 3. So here's the first groups of 3, second group of 3, third group of 3, and then I know that in our language we have a termination of our phrases with a punctuation, a period, and so I can discount everything that comes after. And then you've just deciphered the actual encoded phrase by finding the start and the end of this sequence of 3s, right? These codons essentially, and now we have our sentence. So this is this idea of reading frames. For mRNA, in this example here, you can see that there are potentially 3 different reading frames. So the start, where you start reading these things or start grouping these nucleotides into groups of 3, you can start at the very first nucleotide here, and if you were to start grouping these into groups of 3 nucleotides in a row to form codons, then your reading frame would look like this. This is your first reading frame. And then if you were to go back to that chart we just looked at and look up each of these codons, you would find this sequence of amino acids as the polypeptide product. However, if we were to discount that first amino or that first nucleotide and started reading at that second nucleotide and grouped these guys into groups of 3, then this would be our second reading frame, and it would look like this with these pink blocks as our codons. And then again, if you were to look these up on the codon chart, you would find that these are the associated amino acids, so this would be your new sequence. And note how completely different reading frame 1 amino acid sequence is from reading frame 2's amino acid sequence. And then again, if we were to go back up to that mRNA sequence and just discount the first 2 nucleotides and start grouping with the third one, where this is your new reading frame or reading frame 3, again, you would have a completely different set of codons that then would have a completely different encoded polypeptide product. So it becomes vital to really understand where you begin reading or where you begin grouping these sets of 3. Okay, any questions on those concepts there before we jump into the actual translational event? No questions yet? Okay. As mentioned, these are the key ingredients to the complex, the ribosomal complex. The ribosome itself is the site. It's like the machinery, the factory. And the transfer RNA molecules are the key intermediates that come in with the amino acids and bind complementary to mRNA. And I am going to go over this, even though this is also in my video, because you kind of need to know this in order to do translation. These transfer RNA molecules are super unique. They always form this cloverleaf-like structure because of their internal folding. Remember, this is transfer RNA, so it's just an RNA, single-stranded RNA molecule that folds in this really unique formation. And importantly, there are 2 key parts on this transfer RNA molecule that become relevant to our discussion here. The first is this part called the anticodon. The anticodon is a region of that transfer molecule that is provides 3 nucleotides in a row that are complementary to mRNA nucleotides. So this is also RNA. So you'll always find ribosomal nucleic acid here. In other words, you'll find uracils here instead of thymines. And now we have these rules of binding. So if you have GAC, for example, on mRNA, the transfer RNA that has the complementary nucleotides, or CUG, right, the complementary nucleotides to this codon, this transfer molecule can come and bind to the mRNA here. And this particular transfer molecule that has the CUG anticodon happens to associate with an aspartate. And so this is actually the link between mRNA transcript and amino acid. It's these transfer RNA molecules. So there are just as many transfer RNA molecules as there are series of 3 codons. In other words, minus the 3 stop codons, there are 61 possible transfer RNA molecules, all with those unique anticodons, right? Those unique series of 3 nucleotides in a row. And all of them have a very specific amino acid associated with them. And so when the mRNA transcript is in the ribosomal complex, the purpose of the ribosome is to allow the docking of these transfer RNA molecules with their designated matching or complementary anticodons that bring in those amino acids. And then when multiple transfer RNA molecules are together, then these amino acids can bind together. Let's see how this whole process works. This is a really loose look. I really don't like this picture too much, but it does give you sort of this conceptual understanding. Now note that these gaps that you see here are fake, right? You actually don't see gaps between these triplets of nucleotides, but this is just to give you a picture of what this whole thing looks like. Here's our double-stranded DNA molecule again. In this case, the red molecule is our template. That TAC is transcribed into mRNA, so T complements to A's complement to U's, C's complement to G's. AUG happens to be the start of translation, so they've kind of made it easy for you. This is where translation will begin. The first anticodon, or the first tRNA with the matching anticodon, the complementary nucleotides to this AUG, will come and enter into this equation. So here we have adenine, uracil, and guanine. A's are complementary to U's, U's are complementary to A's, and guanines are complementary to cytosines. So this transfer RNA molecule that has that UAC anticodon down here would come and bind, and this happens to have an F-methionine attached to it. So this is now where we see AUG that is associated to methionine that we saw on that mRNA codon chart. And it's through these transfer molecules that bind complementary to the mRNA and that are attached to amino acids. And so here you can just sort of walk through that whole DNA template, find that mRNA, those mRNA codons or the sequence of mRNA, find the anticodons, and then find their associated amino acids. And this is the link between DNA, mRNA, and amino acids. So this whole process begins with a ribosomal binding site where that small subunit comes and docks at the mRNA in a very particular region. That opens up the binding site for the first transfer molecule to come in and attach with complementary anticodons to the first AUG. And this sets up shop. And this is, let me show you this picture first. Right, so here is your mRNA at the bottom. The first thing is that there's a docking station for the small subunit. Initiation will begin when the first transfer molecule bringing methionine in comes in and docks at that first AUG on the mRNA. And once this docking for the first transfer molecule happens, then the large subunit comes and binds, and this is where you can begin the process of translation. So this site on the ribosomal complex is called the A site. A stands for aminoacyl, but you can call it the acceptor site. Either is fine. The A site is, at the very beginning, the A site is open, and a new incoming transfer molecule will come and dock at the A site. And for each of these, the A site and the P site, each of these sits upon one codon on the mRNA. So it's this perfect docking where each of these parts of the ribosomal complex docks at just one codon. The P site is called the peptidal site, and this is where that growing peptide chain will be, or the amino acids will bind and then continue their elongation essentially at this P site. E is called the exit site, and you'll see the importance of that here in just a second. So the P site is where that very first transfer molecule will sit, right? Because that's the initiation. but every incoming transfer molecule will come and dock at this A site. Let's see a picture of sort of how this process works. So here is from one textbook. I don't know if it's your current one or not. Again, here is the initiation. Again, the small subunit needs to bind first. It makes a docking station for that first transfer molecule to come in. Always, always that first transfer molecule will have UAC anticodon attached to that F-methionine amino acid, and always will dock at that first AUG. That's the start or initiation of translation. Once this docking of the transfer molecule happens, that large subunit comes and sits down on top of this whole complex, and now you have the beginning of this elongation. So this is the initiation, and now we can have elongation. Elongation occurs where incoming transfer molecules come and dock at that A site or acceptor aminoacyl site, right? So they come and dock here. Again, only the transfer molecule that has the complementary anticodon to the next codon in the mRNA sequence will come and dock here. That transfer molecule has a specific amino acid that it is bound to, and it comes and docks at this station. Now we have two different transfer molecules docked at the P site and the A site, and once this occurs, magic happens. There's an enzyme that comes and forms a peptide bond between the P site amino acid and the A site amino acid. So a bond is formed here, which is shown in this picture. Once this bond is formed, note that this transfer molecule loses its covalent bond with its amino acid, so that bond is broken. This transfer molecule is now essentially empty. It doesn't have an amino acid associated to it anymore. So this transfer molecule needs to leave and go and get another amino acid so it can continue this process. And something called translocation occurs where the entire ribosomal complex, the whole thing, will move down one codon spot on that mRNA transcript. The transfer molecules actually stay docked to where they are, so they don't move, but the complex itself moves, which allows for everything to essentially shift place. So what was here on the P site, when this ribosome moves down, this guy is empty and it has to leave, so it's going to exit through this E site, which you see here. What was here at the A site now has this growing peptide chain on it, and it will essentially be moved to that P site, which is what you see here. And now we have an open A site that can accept a new transfer molecule. This is elongation, right? Continuous translocation of that whole ribosomal complex down that mRNA, where those transfer molecules are incoming at the A site, bringing their associated amino acid, and only those that have an anticodon that's complementary to the codons will come and dock at that station. I know that's a mouthful, but that's why I recorded this video elsewhere for you, so you can go and watch that again. There's also some videos for you that are really, really helpful in watching this process. I do not expect anyone to really understand this completely just by talking it over and looking at pictures. I think a video is really important here. We're not going to have time to do that right now, but I just wanted to walk you through this process of elongation just verbally so you can kind of see this association between the mRNA transcript and the tRNA anticodons. Now this process will continue all the way down that mRNA transcript until at the A site, a stop codon is reached. And remember, there's 3 different codons that encode for a termination or a stop codon. There's a particular enzyme that will come in and essentially dock here at that stop codon that will help the whole thing disassociate. So everything falls off, the small subunit, the large subunit. The last transfer molecule is cleaved from that growing polypeptide chain, and then you have this free polypeptide chain that can then be further modified through post-translational modifications into a final protein or enzyme. And that's the whole process. This is also from your book. I just don't like it very much. So you can go through that process there. Right, again, the termination is at that stop codon where release factors will break that bond, and there is a complete polypeptide product. Just so you know, the key difference between eukaryotic and prokaryotic transcription location. So in eukaryotic cells, transcription occurs in the nucleus and translation occurs in the cytoplasm or at the rough endoplasmic reticulum, versus prokaryotic cells, they don't have those different places, so everything can occur simultaneously. And this allows for very, very rapid protein production, which is also why prokaryotic cells divide so much faster than eukaryotic cells. You can see here that even during the process of transcription, as soon as that ribosomal docking site is available on that transcript, it's not bound to that DNA anymore, then the first ribosomal complex can come and form and begin the process of translation immediately. And as soon as that first ribosomal complex passes that whole first ribosomal docking station, another ribosome can come and dock here as well to form something called a polyribosomal complex. So this just provides a massively rapid transcription, translation, or protein synthesis for prokaryotic cells. Okay, so what I really want to do is get you to this part here where we can focus on the shorthand transcription, translation of an mRNA or of double-stranded DNA, and then also using two different types of codon charts to really help you conceptualize this understanding. So this question here, if you've watched the video, I got you guys started on this one, but I want to see how far you can get on this one together. We'll do this one here. So using an mRNA codon chart, determine the mRNA transcript and the translated protein amino acid sequence from the following DNA template. So provided for you with the mRNA codon chart that we looked at before. This is also from your textbook. This is the double-stranded DNA molecule. If you look at it, it should be the same one that we looked at before, but you can start afresh if you want to. To do this, let me take you for just a second over to a whiteboard and I'll show you sort of my rules of transcription translation that I think will be helpful before you jump into this process. If you've already seen my video, then you already have these ones down, but if you haven't yet, then this should be helpful for you. All right, so when you're doing transcription translation, if you follow these simple steps, you should be able to get the correct, correct transcript and translated polypeptide product every time. So you'll always be starting with a double-stranded DNA molecule or a gene, right? Step number one, find the template. Now we're going to keep it simple in our class, and always the template is going to just be your 3 to 5' oriented DNA strand, because our translated or our transcribed product is always 5 to 3. I'm saying we're keeping it simple because there's lots of examples in nature that things are switched around, but we'll keep it simple. Once you find the template, then you transcribe. Right, so you just find that transcript just like we did with our last discussion question. Now that you have that transcript, you're ready to translate it, right? So step number 3, before you can begin the process of translation, remember that you need to find the reading frame, where you're starting. So find the reading frame by identifying the first codon of the initiation of translation, which is AUG. So here, find the first AUG or start codon. Now I want to warn you, and the reason I'm emphasizing this is because I see so many times students do this. Let's say that this was your sequence. Whoops, I didn't mean to say T. Okay, let's say that this was your sequence of nucleotides and you're at step number 3, right? This is your sequence of mRNA and you're trying to find your first AUG. Now I warn you, if you try to do it this way, where you group the first set of 3, and you're like, nope, that's not AUG. Nope, that's not AUG. Nope, that's not AUG. And you continue down this way, you've actually just missed that initiation, right? Do not start grouping or don't start reading at your first nucleotide with groups of 3. Instead, do this like a word search. So you just go nucleotide at a time. A, C, G, G, A, U, G. Well, if you do it that way, you've just found it, right? Here's your first AUG. And this begins your reading frame. And then from here, you group all nucleotides together into codons. So from here, then group into codons. You guys understand the difference there, hopefully? So don't start don't look for your AUGs by grouping in groups of 3 and finding it that way. That is not how you do it. You just work through that whole mRNA transcript until you find the first group of AUG and then start there as your reading frame. Okay, so once we've reached this point, now you have to ask yourself, which type of codon chart are you using? So from here, I'll draw this in a different color. If you're using If you're using an mRNA codon chart, then you move on to step number 4. This is the easy part. Look up your codons on the chart and codons, find the amino acids, and you're done. Okay, so if you have an mRNA codon chart, after you've found that first AUG, you've grouped everything into codons, you simply have to go to that codon chart and look up each of those codons, write down the amino acids in the sequence, and you're done. You found that translated polypeptide product. However, if I provide you with something different, which of course I will. If, however, you are using a transfer RNA codon chart, anti-codon chart, then you have an extra step that is needed here. Then your step number 4 is to find the associated anti-codons. Find the associated anti-codons. In other words, if your first codon, of course, is AUG, then you would say, well, my first transfer RNA anticodon would be UAC, right? So find your anticodons, write them down. You don't need to worry about writing out strand orientation for this one. Once you find your anticodons, then you look up the anticodons in the anticodon chart and find the associated amino acids. And then you're done. Okay, so this, after step number 3, where you find that AUG and you associate or you make your reading frame, grouping all of those codons, you have to make sure you're asking yourself, am I using an mRNA codon chart? In which case I just take those mRNA codons, look them directly up, or am I using a transfer RNA anti-codon chart? So we can go back over to our example. And you can process this one together. Now, I'd like to break you guys out into your breakout groups, but I'm nervous if you guys don't have these slides right in front of you, you're going to lose this sequence. So I guess as long as enough people, there you go. CJ's taking a picture of the screen, that works too. So if you have the ability, let me give you just a second, write down this double-stranded DNA molecule, or if you've already written this down from our last exercise on transcription, you have this down. You do need that genetic code. So get this down, open your textbook if you have that, open your slides if you have that, so you have that genetic chart in front of you, and then I can break you out into breakout groups. Let me just give you a second so you can access that. Okay, what I'm gonna try to do, I'm gonna try to break you out into groups according to your pods. This may just give me, you just might have to give me a second because I've been asked by a few of you guys to do that. So while you are working, oh, I see. Okay. This is taking a long time. Okay, let me get you guys started for the rooms that I have open already. All right, the rest of you guys, I'm just going to get you into different rooms. I need to be working on this. Okay, I'm gonna share a screen with you and I'm gonna delete our whiteboard that we had from earlier. Give me, oh good, it's already deleted. Oh, I guess I didn't even write on it very much. Okay, so this is a review for that genetic engineering activity. And I'll talk to you also at the end of this thing about like I was just overhearing your conversation or her conversation, but talking about the results that I put onto our page for genetic engineering. So let's talk through this concept first, and then I'll describe those results that you saw. Okay, for genetic engineering, and since there's only a small group of you guys, there's only 15 here, so if you have questions as we go along, I don't have my chat window open. Someone just asked me a question there, but just unmute yourself and you can just ask as we go along. And Sean, just hold on to that question. I'll answer it as we go through this whole thing. Okay, so for this exercise for genetic engineering, we had this P-glow plasmid. Let me find where my pen went. And for the sake of clarification, P-glow, this is a plasmid. P is for plasmid. And this is a particular plasmid that is engineered at a biotherapeutic company that produces these plasmids for use in genetic engineering exercises. So that's just the name that they give this one. All of these plasmids have their own site of replication. So they have an ORI site and a couple of really key features that you'll find on here. The way that I draw it is over here on the side. Now, if you watched the video, you would have seen this is called the BLA gene. We're just going to call this ampicillin resistance. Ampicillin resistance is provided by, if you were watching the video, it's called the beta lactamase gene. It's an enzyme, it's an enzyme that encodes for, it's an enzyme that breaks apart the beta lactam ring on ampicillin. So it's what provides this resistance to ampicillin, but we'll just call it ampicillin resistance for our understanding here until we get into our chapter 10, which we'll talk about in a little bit more detail. This is called our selectable marker. So organisms that take up this plasmid are able to grow on a media that has ampicillin on it because this ampicillin resistance factor provides it the ability to tolerate or destroy that ampicillin antibiotic, right? And this allows you to select for successful transformed bacteria or ones that took up this plasmid. Now, in addition to this, in addition to this selectable marker, there's a totally different region of this plasmid that has an operon with genes encoding for arabinose catabolism, right? The ability to break down arabinose. So this is called the arabinose operon. Remember, operons are a collection of related genes. In this case, arabinose is a sugar. This is therefore a catabolic operon. I'll just do arabinose in pink as a designation. It's a sugar. And this operon encodes has genes that encodes for enzymes involved in catabolizing or breaking down arabinose sugar into usable glucose units that can be used for energy production. Now, as mentioned just a little bit ago, all of these operons are regulated at the front by two different things. First is the promoter, P. The promoter is where RNA polymerase is able to bind, okay? So this is the binding site for RNA polymerase. RNA polymerase binds at the promoter, but also at the start of these operons is a regulatory unit called the operator, O, operator. So the operator is a sequence of nucleotides, operator is a nucleotide sequence that is actually the docking station for the repressor. And the repressor is an enzyme that sits right on top of this operator unit under normal circumstances. So this is an enzyme that has an allosteric site and otherwise has an active site that docks to or binds to that operator unit, right? So it sits right here on this DNA. This is the repressor. So Michelle, I'm going to get to that one in just a second, okay? Let me finish talking through this one and then I'll actually show you kind of step by step how we use the selectable marker to identify successful transformants. Let's finish this real quick though. This is called an inducible operon, which if you've watched the lecture videos, you've heard of this already for the lac operon. If you haven't yet, then it'll be a little bit more clear as you get the rest of the way through this chapter. But essentially, inducible operons are in the off position. And it's through this regulation right here that the operator, the region of DNA, is bound by this repressor enzyme. And the enzyme stays bound unless it is regulated by its allosteric site. And it just so happens that arabinose is the allosteric regulator. So when arabinose is present, it will bind allosterically to this repressor, and this binding induces a conformational change, just as we talked about in our last chapter, so that this enzyme no longer is able to bind to its substrate, which is the operator. So when arabinose is present, it binds to the repressor and removes it. And once you have this repressor removed, then this RNA polymerase is able to transcribe all the genes involved in this arabinose operon. Now, the relevance of this is that in our piglo plasmid, this is where our reporter gene which in our case is the green fluorescent protein, or GFP. This is where GFP is fused. Sometimes these genes are taken out, so there actually are no genes for the arabinose catabolism, but sometimes they're still in there. Now, again, what's the purpose of this? Well, under normal circumstances, genetic engineering, the whole purpose is to express a gene of interest, like insulin, that you take out of one organism and you put into what we call an expression vector, another recipient organism. In most cases, it's E. coli or a yeast cell. And the way that you would get it in is by putting it here. So this would normally be our gene of interest. Now, in our exercise, we don't have a gene of interest that we've put into piglo, interest, or GOI, gene of interest. But this is the whole point of genetic engineering, and this is why you would use this plasmid. You would normally, as a researcher, put in or add your gene of interest right here, and you would fuse to your gene of interest this reporter gene. And all of this would be under the control of the arabinose operon, where arabinose will turn on the expression of all of the genes underneath its control, including your gene of interest and your reporter gene. The function of the reporter gene, and there's many different types of reporter genes that can be used. This one is cool because it's a nice visual, super quick, phenotypic demonstration of successful expression of your gene of interest. Again, in our exercise, we don't have a gene of interest. But under all other genetic engineering exercises, when you're actually doing this experimentally, the whole purpose is to stick a gene of interest into this piglo plasmid and get it into your new cell, your E. coli, so the E. coli starts producing massive quantities of your gene or of your protein product. Instead of going through each of the colonies that show up on your transformed plate, which we'll talk about in just a second, and trying to identify your protein in there, the reporter gene just gives you a super quick and dirty visual demonstration that your gene of interest is being expressed into a protein. So that's what this reporter gene is. Now let's look at what the actual exercise of that genetic engineering protocol we had for you, if you'd have done it in class, and how each of these two components on that plasmid are visualized onto media, what they mean. So in our exercise, we had two different tubes. We had a positive piglo tube and a negative piglo tube. To both of these, calcium chloride was added or would have been added, CaCl2. This is an ion. This neutralizes the negatively charged outer membrane of E. coli and also the DNA that's in that plasmid. So the DNA is negatively charged also. The outer membrane, that lipopolysaccharide is strongly polarized, it's negatively charged, and it really repels DNA quite substantially. So this helps neutralize everything so that they can actually come together. For both of these tubes in the protocol, E. coli is added. Whoops, sorry, I'm trying to get these color coded here. E. coli is added to both of these tubes, positive and negative tube. After the addition of E. coli, the P-glow plasmid is added only to the positive tube and not to the negative, right? Now, for transformation, we need to induce competency or make these bacteria able to take this stuff up a lot more readily than they normally would. So that's where heat shocking came in. In the laboratory, you would put these guys on ice for 5 minutes, then you would take them and put them into a 37-degree water bath. And what that effectively does is just shocks open these bacterial cells so that they take in those plasmids from the environment. And you put them back on ice again and let them close back up, and then put nutrient broth in there to give them, let them come to equilibration, begin metabolizing so they don't kick those plasmids out right away. So there is something called transformation efficiency where only some of these cells are actually take up this plasmid and others don't, right? So let me give you a couple of examples of what this might look like. So here's a couple of examples of bacterial cells that took up the plasmid and some that don't. Now remember, the negative tube has no P-glow in it at all. So you would not expect to see anything transformed here. For both of these, we plated them on selective and differential media. Okay, so let's look at what this looks like. These are some of the questions we have for you, is to understand what the purpose of this agar does and all of the different ingredients here. For both, the agar is the same. The agar has ampicillin in it, and it has arabinose in it, and it has standard nutrients, right? Ampicillin, arabinose, and standard nutrients. LB is lauryl broth, which are standard nutrients in there. So this allows the growth of organisms that can tolerate ampicillin, and arabinose will allow us to distinguish between those that are expressing genes under the arabinose operon and those that are not. So let's see how these guys will look. Let's start first with the negative tube. These guys have no P-glow that was added, right? But there are E. coli bacteria. When plated on this ampicillin arabinose LB media, do you expect there to be growth or no growth? And I'll ask you first. You can type this out or answer. Is there going to be growth or no growth from this negative P-glow tube on this selective and differential media? All right, we got one input. Anyone else? Growth or no growth? Only one person. Okay, good. Thank you, guys. Thank you. Okay, so there's expected that there's no growth here. We're out of propane. We're out of propane. Yeah. So there's no growth here at all, expected at least, because these bacteria did not receive this P-glow plasmid that has that resistance factor, right? So they don't have the ability to resist ampicillin. So in this case, ampicillin is acting as our selectable ingredient, selective component. And so there will be no growth of organisms that don't have the ability to resist ampicillin. Now, however, what would be the meaning of a colony that grew on our negative P-glow plate? What would that actually tell you? Growth indicates what? What do you think? If you actually had growth on this negative P-glow plate, what would that mean? Yeah, so somehow that means maybe E. coli is naturally ampicillin resistant. That would be a bad thing, because what we're actually using the selectable marker for is to determine those organisms that took up the plasmid from those that did not. But if we see that E. coli had natural resistance, that means that we have to, no matter what it means, we must discount or disregard all results. I'm going to run out of batteries here in a second. Hold on, I need to move myself to my charger. Sorry about that. Okay. Okay, so as mentioned, if you see growth on here, that means you have to discount the results, because what we're trying to use this negative plate for is our control, but we're trying to ensure that E. coli does not have natural ampicillin resistance. Do you have any other ideas of why you might see growth on this plate? More commonly, and there sometimes you do see growth, more commonly it's not because of natural ampicillin resistance, but it's actually user error. So oftentimes someone will accidentally plate the positive plate on the positive tube on the negative plate, which gives you accidental growth here. But because you don't know that, you still have to discount all results, right? So any growth on the negative plate, essentially you just have to throw out the entire exercise and start over again. Sorry, my writing is so bad here. Okay, or maybe contamination. So these are all possibilities of why you might see growth here, but the expectation is you would have no growth because you have ampicillin in the media acting as that selective component. Now let's go back over to this positive plate. On the positive, with the positive tube, we have bacteria that were transformed, but also lots of bacteria that were not. So let's give an example of what each of these things look like. Yes, please. So let's see what this guy would look like if it was actually transformed. That means it took up that plasmid. You would expect, hopefully you understand, you would expect to see growth of that colony on the media. So for the positive plate, growth is equal to ampicillin resistance, which they gain only by the incorporation of that plasmid. Therefore, growth is equal to successful transformation. Again, we know that because hopefully you saw there was no growth on the negative plate and there was growth on the positive plate, right? I think in the exercise that I provided for you, I think I put plates where you had one that had growth but no glowing and one that had growth and glowing. So I think I'll go back in and modify that so you have a plate that has no growth on it too as your negative P glow. I think that was the problem, wasn't it? I think that the plates that I was showing you were a plate that had arabinose in it and a plate that did not have arabinose in it, or something that I'll show you here. Let's say that this P glow positive tube that had bacteria that were transformed and bacteria that were not transformed here, and also bacteria that were transformed, but perhaps they had a mutation event that occurred that rendered that arabinose operon not working anymore. So let's give an example of all three of these things. In this guy right here, this bacteria that did not take up the plasmid, right here, if this, even in the positive P glow tube, if this guy did not take up the plasmid, would you expect to see growth or no growth on the media? Anyone? Good, Laura. Yeah, for this guy, if it did not take up the plasmid, you would expect to see it not growing on this media, again, because there's ampicillin that will kill anything that does not have ampicillin resistance. So as long as we know that there's no growth on this negative plate, then we would expect there not to be any growth of not transformed bacteria. So Michelle, hopefully to answer your question, what this selective media is doing is allowing you to only grow organisms that were successfully transformed, which is why we call this the selectable marker. Ampicillin resistance gene, or the BLA gene, beta-lactamase, allows you to select for only organisms that have this P-glow plasmid and therefore have this ampicillin resistance factor. So it's selectable. It's a selectable marker because it allows you to select for only successfully transformed bacteria on this selective media. Let's look at one more possibility here. Let's say that this guy here, it is transformed. So it took up that plasmid and we saw growth. But we want to differentiate between organisms that are successfully expressing our gene of interest. This doesn't happen in every transformation. So sometimes there can be errors that occur during replication. Perhaps there's a mutation that happened in this repressor or somewhere along this operon. Or perhaps somehow there was an error in this whole thing got shut down somehow. There's lots of possibilities of this. So even if this plasmid got incorporated, you don't know if that gene of interest is being expressed or not. So this is where that glowing comes in. With this plate, you would then put it under UV light, right? Where's my pen? There we go. You would then expose it to UV light. And UV, with the addition of arabinose, remember that arabinose will remove this repressor and allow for expression of anything that's under the control of this operon. So as long as arabinose is present, we can then differentiate between organisms that are expressing this gene of interest by looking for that green fluorescent protein, and organisms that are not expressing this gene of interest because they will not be glowing. So even if they have this plasmid, they may or may not be expressing this gene of interest, and therefore may or may not be fluorescing green under UV light. So in this example, let's say we expose this plate to UV light. This guy was glowing, and this guy was not. In this case, we can differentiate between organisms that are successfully expressing the gene of interest versus those that are not. So glowing under UV light indicates successful gene expression of your gene of interest. Of course, we don't have that, so we're just looking for that reporter gene of the reporter gene. And those that are not glowing but are still growing on that plate, this would tell you that these guys have no gene expression of the reporter gene, right? They might still have the plasmid, but they're not expressing the reporter gene. Now there's one other possibility that you might see on this positive plate. And these are these little tiny guys that you'll see pop up right next to these successfully transformed organisms, colonies. These we call satellite colonies. Which is just an opportunistic. These guys are not transformed. They did not receive that plasmid, but that beta-lactamase ampicillin resistance enzyme is an exoenzyme that's secreted out, and anywhere where that bacterial colony is growing, it's going to destroy the ampicillin all around that media. So you might have these little opportunistic E. coli or bacteria that pop up in essentially the shadow of these successfully transformed colonies, where there is no more ampicillin to kill the bacteria. They're usually much tinier because they've got none of the stuff up here, and they grow late in the game because they have to wait for that ampicillin to be destroyed. So you might see something like this. So these are three possible things that you might visualize on that ampicillin arabinose LB plate. The ampicillin, again, is the selective component. The arabinose is the differential component because it allows you to differentiate between organisms that are successfully expressing that reporter gene or not expressing the reporter gene, right? So this is the differential component. Of the media. And so you can see some differences there. Okay, do we have questions on this stuff here? Jeff says, is this the info from the YouTube video? Yes. No, this is the same information. I just write it down a little bit differently so you can see essentially the procedure of this genetic engineering exercise next to what the plasmid looks like. So in that video, that YouTube video that I posted on this genetic engineering exercise page, it's really just talking through that P-glow plasmid itself and all these different components of it, and then sort of how it's visualized on the media. And then the other video I posted is the actual exercise itself of what it would look like when you're going through it. So this is sort of just trying to wrap up both of these things together to show you how the plasmid is used in this exercise and how it's visualized on this media. So what I'll do is go in and I'll add one more plate to that genetic engineering exercise tonight so that you have a clear demonstration of what an actual negative plate looks like. And perhaps you can use the plate that has growth but no glowing and explain the results based on what we talked about here, because that is one possibility of a result that you might see there. Any other questions? Anyone? You're welcome to unmute yourself and ask me a question if you have one. Okay, if there's no questions, what I'm gonna do is I have this drawn out very nicely, not me, one of my other students has drawn this out really beautifully, so I'll post that one as soon as I go eat my dinner. I'm gonna post that one for you, and I'll post that negative plate for you too, so you have that to see. Hopefully that'll give you all the information that you need. And I did remember to push record, so I recorded this for you guys too, so I'll post this stuff all this evening on Canvas. And I think this stuff is due tomorrow by midnight, so hopefully you can get that in. If you got no more questions, then that's it for this, and I'll talk to you guys again on Tuesday. Okay. Ciao, everyone.
Good question, Dr.
Oh yeah, sure, Shirley.
Sorry. If I already turned mine in, do I have to redo it if you're saying that you're going to add on?
You don't have to redo it. No, don't worry about it. As long as you have your understand, that's Laura. As long as you understand what you wrote about, that's fine. You can keep it as is, but I'll post that negative plate anyway for anyone who wants to have that clarification in there.
Okay, thank you.
Sure. All right. It looks like I think we're all done here.
Sorry, if I can just ask one more quick question.
Sure, no problem.
When we were doing in class earlier, I had asked, I was like, I don't get it. And I was talking more about the discussion question when you were doing, it says using tRNA with a chart. What were we using that for as opposed to the other chart that we were using, the green one, the green and blue?
Yeah, so the tRNA anticodon chart is where you remember that list of like the steps to get transcription, translation, 1, 2, 3, and then if you're using mRNA codon chart versus if you're using tRNA codon chart. Right. So if you're using a tRNA anticodon chart, essentially you'll need to find that tRNA anticodon, right? What that anticodon sequence would look like on that transfer molecule. And then the tRNA anticodon chart that I have for you is actually those anticodons that have their associated amino acids. So it's just one extra step in sort of the figuring out shorthand of your translated protein product versus the other chart, the blue and green one, that's an mRNA codon chart. And that one, you don't have to find those tRNA anticodons. You simply go transcribe and then find the AUG and then go directly to that mRNA codon chart. Essentially, it's like a super shortcut to get to the associated amino acid product using the mRNA codon chart. And also that's what everyone uses across the entire world. So that's the more common use, is just going from mRNA, find that AUG, find your reading frames, and then just go directly to the mRNA codon chart. But the transfer RNA codon chart, it's an anticodon. So you have to find from your mRNA codons, after you identify AUG and group them all, you find the anticodons, so the complementary codons to those ones, and then you go and look them up on that transfer RNA codon chart.
Like when you guys were saying like AUG, I was looking at it at the actual transfer codon chart, I was looking at AUG, and if I find
not.
that,
So
okay.
if you're, yeah, so if you're, for example, yeah, for that first codon is AUG, but AUG is in the mRNA. So if you were to take AUG and look up the amino acid, you have to look it up on the mRNA codon chart, and you would find AUG is associated with methionine. If you take AUG and look it up on the tRNA anticodon chart, it's not methionine. There's a different amino acid that AUG encodes, because that is talking, that transfer RNA codon chart is actually the anticodons, not the codons. So you would actually take the AUG from mRNA and then find the complementary anticodon, which would be UAC. And then if you take UAC and look that one up in the transfer RNA codon chart, then you would find that that's associated with a methionine.
Oh, I see it. So then I would have to take the actual, okay. I think I get it.
So you should, at the end of using the transfer RNA anticodon chart, as long as you're finding those anticodons first and then looking them up, you're going to have exactly the same answer if you have the same double-stranded DNA molecule that you're starting with. If you work through an mRNA codon chart versus a transfer RNA anticodon chart, as long as you do that one extra step to find the transfer RNA anticodons first, and then you can compare them, and it should be, the answers at least should be exactly the same.
The amino acids should be the same.
Yes.
Okay. That kind of makes a little bit more sense. I know I'm just seeing you, but okay. I'm looking at the charts as we're talking, and I'm like, okay, I'm gonna get it, but I don't know.
And then if you watch the videos, I think I do go over this one, and I do both of those ones again.
Okay.
So yeah,
I haven't had the time because I was trying to get all my lab stuff done.
Okay.
I kind of did it backwards. Sorry.
Okay, no problem. At least you'll have that on there, so you can watch those sections.
Yeah, I've been watching those videos like a lot. All right, thank you so much.
You're welcome. Thanks.
So we find the tRNA and then the mRNA is already spelled out for us, is what you're saying?
No, it just depends on which of those charts you're working with. So in both cases, you'll start with a double-stranded DNA molecule. So you have to go through steps 1, 2, and 3, right? Those steps that we wrote out. So you still have to find the template, find the transcript, find the first AUG, and group those codons accordingly. And then you have to decide if you're working with an mRNA codon chart or a tRNA anticodon chart to determine what your next step is. So that's why I wrote, like, on our step-by-step, steps 1, 2, and 3 are always the same. Find the template DNA, find the transcript, find that first AUG, and group your reading frames. And then from there, if you're working with an mRNA codon chart, all you got to do is take each of those mRNA codons and look them up on your mRNA codon chart and write out the sequence of amino acids. If you're working with a transfer RNA or tRNA codon chart, you have to take all of those codons that you identified in mRNA and then find their complementary anticodons, write them out, and then look those up on the transfer RNA anticodon chart, and then you'll have your amino acid sequence. So it's just adding one extra step if you're using your transfer RNA anticodon chart versus your mRNA codon chart. So you just have to know which chart you're using to figure out which steps you need to go through.
Thank you.
Yep. Rahel or Chikosi, either of you guys have questions?
Yeah, I just have a quick question. Can you hear me?
Yeah. Mm-hmm.
Oh, what's the best way to submit our labs? Like, would you rather have us take a picture or just do it in the document, like in the
That's totally up to you. If you have already written things out by hand, it's probably easier for you to just take pictures and upload them. If you haven't done anything yet and you just want to put them on the computer, as long as you can still write the formatting as we would, normally have in your lab notebook, that's totally fine, and you can submit that one.
Okay, so picture's fine, just taking a picture of it
and totally posting.
Yep. Okay, perfect. That's my only question. Thank you.
You're welcome.
I have one more question. I'm so sorry.
Sure.
So the
Don't apologize.
discussion questions, I know you were making worksheets on their own. I actually put it into my lab. Like, I made it part of my lab.
That's fine, but I do have 2 different places on Canvas to submit those things. So just make sure you can submit all that stuff as part of your lab notebook. But if you could just make a separate document of your discussion, your worksheet thing, and then submit that under that worksheet. It's just hard for me to keep track of all you guys. And if
no, you're fine. I actually just turned in my lab twice, but I will.
Oh, that's totally fine too. I don't mind. As long as you've submitted it under both spots and you have it all in there, that's fine.
Okay, you're just gonna see
I just can't go back and forth and trying to find it different places, but as long as you've submitted it in both
Okay.
places, that's totally fine.
But from this point on, I will then just do worksheets on worksheets and then
I
the
don't care how you do it. As long as you are submitting it in both places, it doesn't matter to me.
sounds good.
Thank you.
Thank you.
Thank you, Professor. I was only listening.
Okay. No
Okay.
questions from you?
No question for now. Thank you so
Okay.
much.
You're
Okay.
welcome.
Okay, bye-bye.
Okay, ciao, everyone.